Welcome to our big biology bash bonanza. Good morning, ladies and gents. Today we're studying DNA replication. Here are our contestants. My name's Matthew, and I'm the best at biology.
I'm Jessie, and I'm gonna fail this thing.
I'm Jamie, and I'm gonna cry because I don't know anything.
Can someone explain what is happening in this picture?
The first model is the conservative model. This model leaves the original template and produces a copy of two new strands containing all new DNA base pairs. This theory is proven to be untrue. I'm not so sure about the other models.
Well, that's an amazing answer. 50 points. Can someone else explain some of these other diagrams?
The second diagram shows the semi-conservative theory. It produces two copies, each containing one of the original strands and a new strand.
Wow, I'm flabbergasted by that answer. 100 points. Finally, can someone explain this model? Yes, this final model is the dispersive model. This model has random scattering of daughter and parent DNA, and it's also proven to be untrue. Since it's random scattering, it can't necessarily be the correct genetic sequence. Since all cells in the body have the same DNA, and this theory is pure randomness, there's no way each set of DNA could be the same. For example, one strand could be 99% parent DNA and 1% daughter DNA, and the other strand could be 43% parent and 57% daughter. That is one great explanation. You're getting 75 points for yourself. Why is the semi-conservative theory correct?
The semi-conservative theory is correct because DNA molecules were prepared using the heavier 15 nitrogen and then induced to replicate in the presence of the lighter 14 nitrogen, which are on the normal DNA. Then the samples were separated via centrifugation to determine the composition of DNA in the replicated molecules, and the test tubes showed that the DNA had densities that matched the semi-conservative model.
Well, this seems like you know your stuff. 100 points. Can anyone explain the initiation phase?
The initiation phase starts with DNA helicase binding to oocytes, which are replication origins. Then DNA gyrase stabilizes the DNA strand and relieves the supercoiling caused by unzipping. Single-stranded binding proteins stabilize the DNA by preventing the base pairs from coming back together.
Wow, that was extravagant. 100 points. Will anybody explain the priming phase?
In the priming phase, DNA primase lays down RNA base pairs in a 5' to 3' direction on the new strand of DNA. RNA primers are needed for DNA polymerase III to attach to.
Well, that was a concise, correct answer. Since it was so simple, only 50 points. The elongation phase needs an explanation. Who's up? DNA polymerase III creates new strands from parental template strands. Then free nucleotides with a phosphate 3 group align with their complementary pair. DNA polymerase III splits the phosphates from the free nucleotides and uses the energy to connect the nucleotide to the new strand. DNA polymerase III joins the 3' end and attaches the free nucleotides in a 5' to 3' direction. DNA is always anti-parallel, so DNA polymerase III will always move in opposite directions. We know the leading strand moves towards a replication fork and it is continuous, whereas the lagging strand moves away from the replication fork and it uses Okazaki fragments. The lagging strand has multiple RNA primers. RNA primers are needed because DNA polymerase III needs 3' OH. And DNA polymerase I removes RNA primers and adds DNA nucleotides. Then DNA ligase makes phosphodiester bonds to fill in the gaps between the Okazaki fragments. Finally, topoisomerase II releases the strain on the parent DNA being unwound. Well, that was a perfect answer, 100 points. Now can someone explain the proofreading phase?
DNA polymerase enzymes play an important role of proofreading and correcting errors as they assemble new DNA strands. These errors are usually base pair mismatches that inhibits hydrogen bonds from forming, and these mismatched base pairs are lumpy instead of a smooth double helix. DNA polymerase 1 and 3 run along newly synthesized DNA, checking for base pair mismatches and cuts mismatched bases and adds the correct replacement. DNA ligase catalyzes phosphodiester bonds to seal the gap in the DNA backbone.
Wowzers. Phenomenal. Great work. 75 points. Can anyone explain this last image?
That diagram shows telomeres. RNA primers are never placed at the exact end of a chromosome, so DNA will be lost, causing shorter chromosomes after every replication. Eukaryotic chromosomes have protective caps, called telomeres, on each end of a repetitive sequence, and loss of telomeres causes loss of coding DNA, suggested as a reason for aging. Immortal cells have an active telomerase enzyme that then adds back to telomeres.
Great answer. 100 points again to one of our smartest contestants.
Did you know that overactive telomerase is a characteristic of cancer?
What a smart cookie. 5 bonus points. Time to announce the winner. Jamie has 205 points, Matthew has a low 175 points, and Jesse has a whopping 375 points. It is clear to say that she may be the best contestant we've ever seen. She wins a prize of a 100% final grade courtesy of our sponsors, Mr. Hundle and Mr. Suku.
Oh my god, I just won!
No!
How did I do so bad?